Okay. There we go. It seems like our webinar has officially started. We're waiting for one of our speakers to join. There was a bit of a glitch, it seems. Didn't receive the link in time. So we're hoping that Michelle Katusele from PNG will join us soon. Really keeping fingers crossed, a pretty important presentation that we'd like to hear. But in the meantime, a very warm welcome to you, colleagues from across Asia Pacific, and indeed from many other parts of the world. Thank you sincerely for setting aside time to join this APMEN Tech Talk webinar. Our webinar today is going to focus on insecticide resistance, monitoring and management. Malaria control, as you well know, is based overwhelmingly on vector control. And since the 1990s, vector control has been based very largely on insecticide-treated bed nets, ITNs. That worked extremely well initially. In fact, ITNs were responsible for about 68% of the reduction in malaria clinical cases between 2000 and 2015. That translates to roughly 600 million cases averted. But since 2015, as you also well know, malaria reduction has stalled and insecticide resistance has been a major contributor to the stall. So our webinars today will look at different aspects of this insecticide resistance in Asia Pacific, and I'm sure we will all find value in these presentations. From all of us, a sincere thanks to our speakers for your time and efforts to share your expertise with us today. So briefly, some comments on the webinar process. We will have 3 PowerPoint presentations, all back to back. This will be followed by the usual question and answer session. Our audience members can submit written questions during the presentations. Please use the question box, not the chat box, please. I also want to suggest precede your question with the name of the panel member you would like to respond to your question. For example, Michelle, and then pose your question. After the Q&A session, there's a very short poll. It's quick, 1 minute of your time, but it really helps us, and I'd be grateful if you can just quickly tick the boxes. It gives us some idea of what we're doing right, what we can improve on, and so on. Please note that this webinar is being recorded and it will be made available on the APMEN and ORIN websites within a day or so. And the details of these sites will be made available on your screens. So, Dr. Borne, or someone, can you tell me if Michelle is with us or hasn't been able to join?
And not yet, Dr. Liu.
Oopsie, all right.
Look, I'm happy to go first if you like.
I was going to push your button and ask if you wouldn't mind, Nigel. So let me introduce you, and thank you for being willing to jump straight in. Our first speaker, therefore, will be Professor Nigel Beebe, who will talk on the topic of behavioral resistance in response to insecticide pressure in the Papua New Guinea region. Nigel has been working in the School of Biological Sciences at the University of Queensland, but with a 50% position at the CSIRO Health and Biosecurity Unit. His work in vector biology, and more specifically the molecular evolution and genetics of mosquitoes in the Indo-Pacific region, delivers fundamental knowledge into the role that mosquitoes play in mosquito-borne disease. And we all thank you for finding time to share some of your insights with us, Nigel. Many thanks. Over to you, Nigel. Thank you.
Okay. Yes, can you stop sharing screen? And I'll do. Okay. Share screen. Let's see if I can find it again.
Willem, can I ask you in the meantime, send Michelle a message telling her we're live already, if you don't mind. Thank you.
I did send. Thank you.
Thank you.
Look, yeah, thanks, Leo, and welcome everybody. Greetings. Yes, I'm Nigel Beebe from the University of Queensland, and the CSIRO, it's a government science institution for industry in Australia. On the image you're looking at on the right-hand side is the University of Queensland, and you can walk across that bridge, and in 15 minutes you'll be at the Boggo Road Eco Science Precinct, where the CSIRO is. Look, I do work in the area of molecular evolution. Doesn't want to. Here we go. I do work in the area of molecular evolution and genetics, but I'm not going to be talking about that. What I want to do is talk about behavioural resistance and maybe just to throw an overview around and a bit of history about in our part of the world about behavioural resistance and what we've seen so far. Okay, so the main malaria vectors of this region are the Anopheles punculatus group. They exist from the Moluccas through Indonesia all the way down to Vanuatu. And initially there were three morphological species, Anopheles ferrati, Anopheles punculatus, and Anopheles coliensis. But now we have an array of about 13 cryptic species. The molecular genetics tools that we developed to identify them has identified 8 versions of Anopheles ferrati, morphologically identical. Potentially 3 versions of Anopheles coliensis, Anopheles punculatus, and species near punculatus and chloe. And the molecular diagnostics actually I developed last century, they're very old now, but we still use them, are still able to separate these species. And over the last 25 years, working with other groups, including the Australian Defence Force and even PACMOSI, we've been able to identify the major malaria vectors from the minor malaria vectors in this group, and we're down to Anopheles ferrati, Anopheles hinesorum used to be called ferrati 2, Anopheles ferrati 4, Anopheles ferrati 8, coliensis. Again, we don't know which species within that because the identification of these species is relatively new, and Anopheles punculatus. So being the primary malaria vectors, I don't want to also detract from, there are some secondary vectors that are in isolated pockets throughout Papua New Guinea or in New Guinea itself. And these are Anopheles bancrofti group and the Longirostris group. And we have molecular diagnostics for all of those species, and they're really based on the same premise where you need to, all you have to do is PCR amplify up the ITS2 and cut it with a restriction enzyme. The same enzyme works across all 3 species. So if you do get a chance to be studying these mosquitoes, the bancrofti and Longirostris group, it is worth trying to get down to the species level. Just a little advertisement there. What we have found over the last 25 years, and these are some maps of the distributions of this from the sampling we've been doing over the last 25, 30 years, of showing Anopheles ferrati and hinesorum, punculatus, coliensis, and ferrati 4 and ferrati 8. You can see they have very different distributions. They have different biologies and ecologies, and they will adapt differently to vector control. So we need to be able to identify those species quite well. But probably just do a little bit of brief history. What do we know about biochemical resistance in the punculatus group? And so I dug into some of the old literature, and I understood that in the 1940s and 50s, DDT was actually used as a larvicide for breeding sites in PNG, and it had short-term effectiveness. But think about larvae swimming around in dilute DDT and what that would be selecting for in regard to fitness. And from the late 1950s to the late 1970s, DDT-IRS was used in PNG and probably through the Solomon Islands as well. When it comes to documenting resistance, DDT resistance was documented in Anopheles coliensis in northern Papua province. This came from Sleuth's PhD, which can be sometimes hard to find. And in 1988, 99, there was a study that showed that 30% of Anopheles coliensis in northwest Papuan Peninsula was DDT resistant, and that was Mikey Bangs and colleagues' paper that came out in 1993. Also dug in and could find that there was dildrin resistance documented in Anopheles ferrati 1 from Honiara in the Solomon Islands. So if we push forward to sort of the more recent times, we're using pyrethroids now. Going through the literature, we see no pyrethroid resistance documented in the punculatus group members. Okay, there's been a couple of papers that are down there. But interestingly, Rebecca Vinnett in the last APMEN Tech talk presented some suspected pyrethroid resistance emerging in Anopheles punculatus and Anopheles coliensis, which was really interesting, and some suspected organochloride resistance in ferrati and punculatus. But that's all I'm going to talk about biochemical resistance. I'm really interested in sort of diving into the area of behavioural resistance, which seems to be more of an issue in our part of the world. First of all, I want to show you a very old study that came from Tony Sweeney's master's thesis. He went to the Cataracts just off Bougainville, where no DDT IRS had ever been instigated. And he found that the classic, or maybe the native biting profile of Anopheles ferrati was it started biting just after sunset and it bit all the way through the night. Okay, and we can call it maybe the classic biting profile of Anopheles ferrati. And then in a paper by Brian Taylor in 1975, who was studying not malaria DDT IRS activities in San Cristobal Island, just in the southern part of the Solomon Islands, was following Anopheles ferrati, its behavior and biting during the night, and looking at its outdoor and indoor biting profile. This mosquito, yes, it starts biting just after sunset and it bites through the night, and probably at a decreased intensity later in the night. What was interesting that 2 years later, post the DDT IRS activities, the behavior really changed. It swung to biting preferentially earlier in the evening. And interestingly, Anopheles punctulatus and Anopheles coliensis that were also in the landscape completely disappeared, and coliensis has been very difficult to find in the Solomon Islands since. This change in biting behavior actually broke the back of the malaria eradication campaign. It was being run by the British at the time, and they couldn't shut malaria down, so they pulled out. And this biting behavior is maintained today, which is even more interesting because evolutionary theory would suggest it would go back to the classic biting behavior, but it hasn't. It stayed as an early evening biting preference. And this wasn't just documented in San Cristobal and Solomon Islands. This was seen in quite a few places with the DDT IRS that was happening in the 1960s and 1970s. So it wasn't unique to just one population or one island. So there's a little bit of history there. And what I want to do now is take us forward in time to some more recent studies that we are utilizing now because we're rolling out, insecticide-treated nets. And so a study by Bob Cooper and Steve Francis in 2002, looking at Anopheles ferrati, identified the biting profile as being more of that classic ferrati biting profile, even though that the region of Booker and Bougainville were undergoing 2 sprays a year from 1960 to the early 1980s. I was checking my notes, but the behavior didn't change. Well, it may have changed, but it's reverted back. Russell et al. In 2016, we all went into the island of Gela, a study site called Halata. When we turned up, the biting profile was primarily early evening. And over the process of about 7 years of studies, we found that the bed nets are actually fortifying or strengthening that early evening biting phenotype in that part in that study region. A similar study by Hugo Boguera and colleagues working in Tuamotu, the most eastern island of the Solomon Islands. When they turned up, they found an early evening biting profile. The nets were rolled out after 2008 and 2010, and in April and December, the biting profile was probably reinforced into more early evening biting. Even it was difficult to glean why the numbers of mosquitoes were down in the April 2010. It may I'll have to go back into the paper. I've forgotten what drove that. Interestingly, Wang Chao was doing a study in Queensland on Anopheles ferrati population, and this is a population that's never experienced indoor residue spraying. It's never experienced insecticides at all. And it had a biting profile that, again, starts just after sunset and travels through the night, and the mosquito bites through the night. You can see that there's a wet season and a dry season, and the dry season numbers of ferrati are much higher than in the wet season. This is a common occurrence with ferrati because during wet seasons and lots of rain, the brackish swamps often get flushed out and the larvae are flushed out into the ocean. Where in the dry season, the breeding sites are more productive. And you might notice that the propensity to be biting earlier in the evening in this study in Queensland does look similar to what's in the Solomon Islands. But our dry season is a much cooler season in Queensland. And the increase in biting preference early in the evening appears to be driven more by a change in temperature. So as the sun sets and the evening cools down, the Anopheles frati biting through the night seems to decrease. So really some interesting biology that's happening in our part of the world. So if we come up into Papua New Guinea, there was a lovely study done in the Wasserman and Madang region by Bennett and colleagues. And this is pre-bednet rollout. So these are the biting profiles of Anopheles frati. We're looking at percent total catch and time of the night, where frati is biting all the way through the night. Anopheles hynesorum has early evening biting activity. It still bites through the night, but most of its feeding happens before 9 o'clock. And same with Anopheles frati 4. It starts quite early in the evening and bites throughout the night. With Anopheles coliensis, it was a little bit different. They actually found some molecular variants in my diagnostic in the Wasser and Madang region of Papua New Guinea. And in a follow-up study, we were actually able to identify that they actually called them the Madang variant, the Wasser variant, and the Madang Wasser variant based on where they were collected. We were able to identify that the Madang Wasser and the Wasser variants were reproductively isolated using microsatellites. We just published that in 2001, I think. But the jury's still out about this variant in the Madang region. But again, coliensis in 2 of these variants are starting to feed very early, quite early in the evening, just after sunset, and they tend to bite through the night. So if we stay in Papua New Guinea, and there are some lovely studies. Lisa Rima and colleagues did this study in northern PNG, looking into several villages and looking at the change in biting profile after bed nets have been rolled in. So we are currently looking at Anopheles frati in 2 villages, and we can see that before the nets were installed, we had a particular biting profile. Again, it started relatively early and it peaked at about 9, 10 o'clock at night. There was a distinct shift in the second year after the bed nets were rolled out. And this also occurred in Murrap as well. These villages are coastal villages where frati is an important vector. In the same study, Anopheles punctulatus, again, traditionally a late night biting mosquito, usually bites after midnight, as we can see in these whisker plots where the median is about 1 o'clock in the morning, and punctulatus usually starts biting after 9 p.m. What we can see in some of these villages, there's a shift to an early evening biting profile. And only in Yaratong village that we see the shift. We can follow it through 3 years, and we can see that shift in the second year seems to move slightly back, which is, to me, relatively encouraging. In this village here, we actually see a median moving to a later night biting preference. And when I looked into the paper, the actual numbers of mosquitoes that were collected for the year 2 was diminished. And this may be more of an artifact, but punctulatus is still maintained as a late night biting species, which is good for the utility of the bed nets. Okay, so another study by Thompson et al. Looking at Anopheles frati 4 in Papua New Guinea, again, before and after the bed nets are rolled out. We have biting profiles in Cocofine and Manuno, and we see that after the bed nets are rolled out, a shift is occurring to an early evening phenotype. This shift in Cocofine seems to punch back a bit to closer to its original biting profile. But in May Uno, that phenotype seems to have maintained an early evening biting profile. So different things are happening in different villages. These villages are actually quite close together. So there's a lot of selection pressure happening on these mosquitoes at the moment. Okay, this is kind of my last data slide, but this is a study by Kevin Bosco, John Kevin Bosco and his colleagues. It's slightly different in its makeup. Again, this is 5 years after the deployment of nets. And we are looking at the biting profile between 6 and 10, 10 and 2, and 2 and 6 in the morning. And we have Anopheles coliensis in 3 villages. And we're seeing slightly different outcomes occurring, where coliensis is still biting through the night, but in Bulau, we see a propensity for this early biting phenotype appearing. Now, we don't know which species within Coliensis this might be, so we might be able to see species-specific differences, but we don't really know. Same with Wasab, the mosquito seems to be biting through the night. We move to Anopheles ferrati, we see this distinctive behavioural change to ferrati biting more frequently early in the evening. Not so obvious in Marap village as well. But what flagged my attention is what is normally a late night biting species, Anopheles punculatus, was identified as biting quite early in the evening between 6 and 10. And usually punculatus is a late evening biter, starting to bite after 9 p.m. and biting through to the late hours of the morning. So that was, to me, a big surprise. So just sort of bringing this conversation all home, the initiation of blood feeding in the punculatus group occurs in many species, okay? We see it in ferrati, and we see a strong selection for early biting phenotype that can sort of get around the control measures of indoor residue spraying and potentially bed nets. We're also seeing that phenotype in Hynosaurum and in Coliensis, depending on which genotypal species you're looking at, we have both an early evening and late evening biting phenotype. This is also seen in Anopheles ferrati before. And Anopheles punculatus, normally a late night biting mosquito, that John Kevin's publication recently makes me think that this selection pressure can also potentially affect Anopheles punculatus to potentially bite earlier in the evening. So this is all food for thought. And hopefully, it'd be great to get some interesting conversation in the discussion forum. Just before I leave, one thing I found to say is that with the Anopheles ferrati early biting phenotype, most of the nulliparous or young mosquitoes are in that cohort that bite early in the evening. So that was an interesting piece of biology as well. So I'm just going to say thanks and stop sharing my screen, and happy to be part of the discussion afterwards.
Sorry, I'm trying to get myself.
That's okay. That's
yeah.
okay.
So Nigel, okay, it says it cannot start your video because host has stopped it, but that's okay. At least you can hear me. Yep. So Nigel, absolutely fascinating presentation there. I find it fascinating that how responsive populations are within a relatively short time span. You were showing results there within 2 years of commencement of a particular intervention, how there's a directional shift, to avoid contact with the bed nets or shift to a safer biting period. All very important to understand and know about, so one can advise people. Not sure it always happens or there's always an appropriate response, but at least so that programs and people are aware of these shifts, and then how to reduce your exposure risk or perhaps bring in appropriate interventions, though our backs are against the wall with outdoor biting. But Nigel, sincere thanks for that. Great to have that presentation.
You're welcome, thanks.
Thank you for that, Nigel. So Wulan, Dr. Borne, is Michelle with us?
Yes, yes.
Michelle, thank you so much. You had me very, very worried. And I really, and I'm sure everybody else is really looking forward to your presentation. So apologies for whatever glitch there was, but thank you for persisting. And we're very glad to have you on board and part of the team again. So I'm going to introduce you, if you're ready, to give your presentation, Michelle. Michelle Katusele is going to inform us about insecticide resistance surveillance in malaria and arbovirus vectors in Papua New Guinea. Michelle is a senior scientific officer at the Papua New Guinea Institute of Medical Research, where she has worked on vector biology for over 10 years. And her main research interest focuses on insecticide resistance monitoring in malaria vectors in PNG and the evaluation of vector control tools within that country. So, Michelle, thank you for your willingness to share your experience. We look forward to your presentation. Over to you.
Thank you so much, Dr. Braack. Could you share my slides, please?
Sure. Popo, Wulan, Dr. Pon, can one of you share?
Yes, I can share. Just a second, please.
Thank you, Popo. Please just bear with us for a few seconds while Popo finds it and uploads.
That's fine. Thank you, and big apologies for joining in late. And thank you, Dr. Braack and APMEN for the invitation.
You're absolutely no need to apologize. These things happen. We're just very happy you were able to join eventually. Thank you, Michelle.
My pleasure. Thank you.
Is that okay?
All right.
You see the screen?
Yes.
Excellent. You must
Thank
just prompt us, Michelle, tell us change slide or so on.
Yes. All right. Thank you so much. So here I'll be presenting our findings in Papua New Guinea on a current ongoing insecticide resistance surveillance program, monitoring malaria and arbovirus vectors. And these data are from 2017 to 2022. Next, please. Going really briefly through the introduction. So it is really important for us to have insecticide resistance surveillance in mosquito vectors, particularly because we mainly depend on insecticide-based vector control tools. So we need to monitor the continued efficacy of these interventions. And furthermore, surveillance data guides us in the selection and application of the most appropriate combinations of products, as well as active ingredients, especially in the light of increased reports of widespread insecticide resistance in mosquito vector populations in many countries. Next, please. So here we're just showing the endemicity of vector-borne diseases in Papua New Guinea. The ones highlighted within the red oblong are the major vector-borne diseases and the vector species that transmit these diseases. So these diseases highlighted are the target species that we target for monitoring insecticide resistance. Next, please. So this slide just shows the a timeline of the different insecticides used in country. In particular for malaria control. So we've used DDT in the 1950s and then permethrin bed nets in the 1980s to deltametrin L-alliance in the 2000s. And in recent years, we've also have some products of alpha cypermethrin L-alliance also distributed to the communities with deltametrin L-alliance. So our major control intervention nationwide are L-alliance, where we have 80% national coverage since 2014. However, in recent years, they've shown that only in a few countries they've maintained coverage at that level. Furthermore, there's localized interventions in small areas or in localized areas, and these are conducted by public or private entities. Next, please. So in Papua New Guinea, we do have data on insecticide resistance for malaria vectors dating back to 2011. And we have shown since then our vector populations to be fully susceptible to pyrethroids. And I should also add that PNG is one of the countries that we still maintain, or we still show fully susceptible vector species to pyrethroids. In terms of Aedes IR status, the information is quite limited. The first report was in 2019 showing Aedes aegypti and Aedes albopictus being DDT resistant, as well as Aedes aegypti being showing paratroid resistance. Next slide, please. So this ongoing program provides updated report on the resistant status of malaria vectors, which is one of the malaria indicators for our National Malaria Control Program. But we also provide report on arbovirus vectors, and the monitoring is conducted across 9 provinces that are in gray stripes on the map, and we test these mosquitoes to 8 insecticides. Next, please. So here, the slide just shows how we go about doing the surveillance. So we do larval sampling where we collect mosquito larvae from known larval habitats of Anophelins and Aedes vectors. In some provinces, we've also conducted ovitraps to collect Aedes larvae. And the mosquitoes are then reared, and we used 2 to 5 day old female mosquitoes in WHO susceptibility bioassays. And after taking 24-hour mortality readings, these mosquitoes are then identified by microscopy to morphospecies level. This is basically because our Anopheles vectors, there are some members that are morphologically similar or cryptic. So at this stage, the mosquitoes are then stored for further analysis. Next, please. So here, it's just showing that from the surveillance from 2007 to 2022, we've also sent subsets of Aedes aegypti and Aedes albopictus to our collaborators at the University of Melbourne, and they've genotyped these mosquitoes for detection of KDM mutations or paratroid resistant target genes in Aedes aegypti and Aedes albopictus. And the mosquitoes were also sent for gene sequencing. Next, please. So onto the results. The slide basically just shows the mosquitoes, the vectors that we tested across the 9 provinces. So here for Anopheles vectors, we tested mostly Anopheles punctulatus in gray and Anopheles farauti in yellow. Next, please. Onto the Aedes vectors, we tested Anopheles Aedes albopictus across all 9 provinces, Aedes aegypti in shaded. Next, please. So the slide here just shows the overview of the resistant profile of the 5 vector species to the 4 different insecticide classes. So very briefly, here we show Aedes aegypti and Aedes albopictus, as well as some members of Anopheles punctulatus resistant to organochlorides. And we also show that Aedes aegypti is resistant to paratroids. Next, please. Looking into more detail at the morphospecies level for malaria vectors, Anopheles farauti morphospecies across all different provinces show susceptibility to all the insecticides that we tested. However, we do see reduced mortality rates to DDT. Next, please. Furthermore, in Anopheles punctulatus, this is similar except in ECP province, or ESP, on the graph, and that shows the Anopheles qualiensis morphospecies to be resistant to DDT. And we also show reduced mortality rates to Lambda cyhalothrin within that province. And this is similar also in Anopheles punctulatus, where they are susceptible to almost all of the insecticides that we tested, except for DDT showing reduced mortality rates. And in West Pacific province, or WSP, we also show that there's a reduced mortality rates to Lambda cyhalothrin. Next, please. So here we're showing resistance profile of the Aedes aegypti and Aedes albopictus. So Aedes aegypti on the top graph shows resistance to all the pyrethroids that we tested, as well as DDT, and showing signs of resistance to Bandioca. In most of the population that we tested, they were fully susceptible to malathion. Resistance profile for Aedes albopictus, however, they are susceptible to most of the insecticides we tested, but more importantly, they are showing resistance to DDT in several of the provinces, and also reduced mortality rates to some of the pyrethroids that we tested. Next, please. So here I'm just showing the KDA mutation that we detected in Aedes aegypti populations in PNG. So the table on the left just shows the 6 different composite genotypes that we detected. So these are mutations that were detected along 3 different codons on the sodium sensitive, voltage sensitive sodium channel of the species. And the graph on the right just basically shows the that these mutations within these tested mosquitoes is widespread throughout those populations. The Aedes aegypti, the evolution of Aedes aegypti, we now have recent data from Tom Smith from the University of Melbourne that shows that these mutations within the country are imported mutations, and rather than these mutations evolving naturally within these populations. Next, please. So here for Anopheles albopictus, looking at the codon 1534 of the same target site, most of the samples that were tested are wild type. However, we do find in very low frequencies mutations where in one province we did detect a single specimen having a homozygous, being a homozygous mutant, yes. Here again, evolution of KDR mutation within Aedes albopictus in PNG, the story remains unclear. So up to this point, the data that I've presented are available. Our manuscript is currently in press. However, there's a preprint available online should you want to get more information. Next, please. So we have also done further work to detect KDR mutations in Anopheles punctulatus vectors. These are preliminary data that I'm showing here. So the table at the top just shows the phenotypic resistant phenotypes and the susceptible phenotypes regardless of species. And here we show that the 89% of these resistant phenotypes carry these heterozygous resistant mutant, and 42% of the susceptible phenotype also carry these heterozygous allele. The table below just basically shows the sample size and the frequency rates of KDR positive samples that we've detected so far, and this is depicted on the maps on the right across the four species that we confirmed by PCR. So all 3 primary vector species of malaria, as well as which is Anopheles show that there are KDR mutants detected within these populations and across the different provinces in the country. Next, please. So the data that I've presented so far is one of the largest overview of insecticide resistance data set currently in PNG. We show that the malaria population remain susceptible to pyrethroids, but they do show resistance to DDT. Now, this opens up a lot of questions about where this DDT exposure is coming in, whether it's historical or through agricultural runoffs, or whether or not the detection of the mutation now within these primary vector populations comparing that resistance either to DDT or lambda-cyhalothrin, or whether or not there is a cross-resistance between the 2 insecticides. And molecular work so far to confirm the conference of of the KDA mutation to DDT resistance. Conferring DDT resistance is still ongoing, and hopefully in the near future we'll be able to provide more information. Furthermore, we confirm from previous reports that Aedes aegypti are pyrethroid and DDT resistant, but we're also showing that there's a possible bendiocarp resistance. And within this population, there are 6 different KDA mutations that are detected, and these are widespread. 3 of these KDA mutations are known to confer pyrethroid resistance within this species. Furthermore, we also confirmed that Aedes albopictus is DDT resistant, and with the detection of very low frequency of KDA mutation within this mosquito species, there might be a possible resistance to pyrethroid in the near future. Next, please. So to conclude, really in Papua New Guinea, we do need to continue monitoring of insecticide resistance for all vector populations. So currently we use WHO susceptibility bioassays. There are other available testing tools available that we also need to look into and use for monitoring to gain a more clear picture of the IR status of all vector populations in the country. Furthermore, in the light of Aedes aegypti having multiple insecticide resistance, there is also a need, especially at provincial level, to have a single regulation body to ensure that control tools are standardized and insecticides are standardized so that we can maintain the available resources that we have to control these vector species. And furthermore, with the detection of resistance to DDT, as well as the multiple resistance in the Aedes vectors, the understanding of the landscape of insecticide within the country is very limited, and it opens up to a lot of questions to understand this landscape, and also looking not only at public health insecticide use, but also across agriculture as well. And finally, it is important for us within the country to build on our capacity facilities for molecular IR surveillance, because these are very important complementary data set to phenotypic IR data. And especially with KDA mutation now detected across our anopheles species, it's important to monitor this the spread of the KDA mutation and develop strategies to mitigate the spread. Next, please. So this brings me to the end of my presentation, and this slide is very important. The data presented today is a culmination of a huge collaborative effort from different organizations across PNG. Thank you so much for the support in ensuring the success of the program. Funding from Global Fund through the Rotarians Against Malaria were instrumental in the administration, logistics, and procurement of the program, as well as through Stripe PNG. And the anopheline molecular work was funded through Wellcome Trust. Thank you so much.
Michelle, you need to be congratulated on the work you've been doing there and the extent and depth of the findings that you've produced thus far. Very happy that you were able to show us some of your findings on Aedes, very important aspect that as well. So lots of information, data to chew on, to think about, and I'm sure there will be some questions for you during the Q&A session. Thank you for that, Michelle. I should just mention, your voice tends to come and go, and there are periods when it's difficult to hear your voice. I'm not sure if your finger was covering the little microphone or not, but just to make you aware that sometimes there's a little bit of an audio problem. Michelle? we could follow the gist of the conversation fine, but sometimes you have to strain a bit to hear. Michelle, again, thank you so much for your presentation. I think there have been...
My pleasure.
Thank you, Michelle. There are some of our audience members that came in a little bit after the initial introduction. Just to mention to you that the webinar is being recorded and it will be made available on a website that has been posted in the chat box. I see some people have been asking questions if the presentations will be available. So the whole webinar will be recorded and it is available on a website that's listed in the chat box. And also for those who came in a bit late, we switched the presentations of Michelle and Nigel. So both those presentations have now taken place. And we move on to Dr. Pradeep Srivastava, who's no stranger to our Asia Pacific vector community. Pradeep has been co-chair of the APMEN VCWG Vector Control Working Group for a good number of years previously, and given multiple presentations as part of the APMEN webinar series. And besides that, he's also presented short courses for us as well. So Pradeep is going to tell us about the new Bhutan insecticide resistance monitoring and management plan as an example, which he developed at the request of and in collaboration with the Bhutan National Malaria Control Program. Dr. Srivastava has worked as scientist and then at senior level in the India National Program for more than 30 years, including as joint director of the National Center for Vector-Borne Disease. He was also the nodal officer for lymphatic filariasis elimination in India. He has been associated with WHO and APMEN for many years to help frame or lead development of documents related to vector control needs assessment, strategic planning, and insecticide resistance monitoring plans, and others. So we thank you for your sustained commitment to malaria elimination efforts across Asia Pacific, Dr. Pradeep. Thank you so much for your time. Over to you, Pradeep.
Thank you, Leo, and I'm really obliged for your opportunity. Unfortunately, I was not in India, but anyway, thanks. It's a good thing. Let me share my screen. Thank you. And while congratulating the previous two speakers, both Nigel and Michelle, that they have given the real findings and strength to my talks, that yes, why resistance monitoring is very, very important and crucial. There may be many questions and all those things. Because you have introduced from the program perspective, before we start the Bhutan presentation, let me take few moments to what is actually required for the program people, because there are many things which are being asked. So is it okay now, visible?
Yes, thank you.
Excellent.
Okay.
Thank you, Pradeep.
So I have just divided my presentation into 6. The first one is, what is insecticide resistance? We must know when we are talking about the insecticide. I'm not going to define all those things, but there are some confusions that the standard definition of insecticide resistance I have already given on that basically the WHO what decides that it's a dose where it will be lethal and then some species. But the more clear definition has come from Insecticide Resistance Advisory Committee, and the theme of this definition is basically that the last point is very, very important, because with this definition, this is very clear that even the susceptibility apparent laboratory bias may not necessarily constitute the resistance in the different, and that is what has been the basis for national programs that only based on insecticide resistance, you do not take the decision for changing the insecticide. I was very happy to go through, so I thought let me show these two definitions for the people who are from the program and they are doing the monitoring or taking the help from the research institutions. So just a glimpse, one slide only, that resistance monitoring is very, very important, and there are two methods primarily. One, we have been doing traditional WHO susceptibility test. I'm not going to explain this. Then there are, if there is a resistance suspected with the clear definition of mortality or moribund or 80 to 80%, 90% below, or 90 to 98% or above 98%, then we have to go for knowing the resistance mechanism. Plus, we have to do some CDC. So we 5 times multiply and then 10 times multiply the dose and then see whether exactly that will substantiate whether you have to sync with epidemiological parameter, because the moment you are going for malaria elimination, you may not have the same parameters applicable for yours. So you have to have the number of the cases, maybe in tens or hundreds, or maybe zero. So how do you take a decision for making to overcome the resistance if at all it's declared? So that is how this insecticide resistance mechanism, tube test and bottle test are to be done. Now, the basic issue of today's talk is management planning. Management planning is, we must understand that resistance is always, it is susceptibles, they are dying, and the resistant genes are surviving, slowly multiplying. And then, but if the pressure is, insecticide pressure is withdrawn, it can change back. It can go back as susceptible. That is how the principle, the whole philosophy of insecticide resistance management, to delay the resistance precipitation or to delay or to overcome the resistant population, how to reduce the numbers, how to reduce the longevity. That is what is the whole episode is. Now, what is basic aim of the resistance management is basically we are trying to prolonging the effectiveness of insecticide, countering the development of resistance. Then we are also in the process of developing alternative tools, but I am afraid that maybe in some places it may be effective, but in areas where you require large quantity of insecticide and there are many multiple agencies, there may be many multiple issues. I have dealt in one slide later. And then national capacity also for insecticide resistance monitoring. One small population, I'm talking especially referring for Indian system, or in a bigger countries like India, it will be little real to manage with one uniform policy. Then generating evidences and mobilizing resources, very important point. Without fund, nothing will move. So just 4 points I have given, that what is resistance mechanism versus management. Basically, resistance mechanism, when we have just heard two talks, that modifying the target site due to KDR gene mutation or affecting which is acetylcholinesterase or VGS channels, or metabolic resistance by detoxifying insecticide, or behavioral resistance or cuticular resistance. These are the major mechanism, resistance mechanism. But how to manage? If there is a KDR mutation, it is all proven fact, then we have to have the rotation of insecticide, rational use of insecticide. How best we can use judicious use of insecticide, and basic thing comes on the integrated vector management, which will be coming later. Then maintenance of doses. Can we, for second, if metabolic resistance, when the dose is increased or dose is regulated, so that the aim is to kill the susceptible or heterozygotes especially, because if you really recall the Mendelian's law, there will be initial phases, there will be heterozygote resistant and susceptible genes. Then for the rotational use of unrelated insecticide is very, very important. And that is what is there, rotation. But there are some inherent problems for the larger areas. And then manipulation or blocking the detoxifying synergist we are using, PVO nets are very, very common nowadays. But again, there are some challenges. So these are the 4 basic mechanism and 4 kinds of management, which we will be focusing on the management issue. This was just courtesy of Iraq. This is if you do not manage the resistance, if you do not properly monitor the insecticide resistance to generate the evidence for management policy, then what is going to happen? Without resistance, if you see the red color, it is just 4 times and another 4 times multiplication factor. It has been taken. It is an hypothetical graph that if one compound A fails, you have to go switch over, and it cost goes around 4 times more. And if B also fails, you switch over to C, it is again 4 times costlier. So if you do like this, yes, you may have some, but if you don't monitor and don't manage this, switching over, you continue with the same compound, you are going for higher cost. This is all hypothetical. There is no data collected on this basis, but this is a trend which has been analyzed, and I'm thankful for IRAC website for this. Then global program, also most of the people who are in the audience, they all know that these are the four principles which has been given, recommended by the WHO and the Global Plan for Insecticide Resistance Management: rotation of insecticide, mosaic, use of mixtures, and combinations of interventions. Now, there are some principles for that. Basic concept is that we have to remove the selection pressure by either by rotation of insecticide. You are just trying to deceive the insect that okay, you are exposed to one, then suddenly you are exposed to one, so they are befooled. And then overpowering the resistance, continuously you go on killing by mixtures or combined intervention so that none is left. So it is but the basic issue, I'll touch on that. So when we want to remove the selection pressure, what is basic aim? Basic aim is to enhance the survival of susceptible. As I showed you in the one graph, that once the pressure is removed, the survival, the reversal phenomena takes place. So susceptible homozygotes, relatively, they will come back, and use of rotation and mosaic basically depends on this theory, because we will try to expose one kind of insecticide, then another kind of insecticide. So the population susceptible homozygotes, they will be it will be delayed, it will not be prolonged. And that is what the philosophy, the basic aim is, that we have to delay the resistance, because the choice is very, very limited. We have to deal within the available insecticide as on date. The another point is very, very important, that when we think of rotation in the resistance management, it is basically the first graph which I showed is very, very important every time, that the susceptible population will come back because of their reproduction rate. And in the absence of insecticide, this susceptible population, when they are coming, they will try to overpower the resistant genes. That is what the whole concept of the rotation principle is. This is one example. This is a good example for the people who are actually in the program, that say, for example, we have not done anything, single insecticide we have been using for continuously. Then another compound we switched over. I have just not shown malathion or organophosphate, pyrethroids, single insecticide. But what will happen if we want to use the rotation of insecticide? You use organophosphate, then you use pyrethroid, then you use carbamate, then you use again pyrethroid, then you. So here the population where we are attacking, they are actually confused against which the resistance will precipitate. So basically the phenomena is confusing. But slightly different from rotation to mosaic. Mosaic is here, suppose one insecticide we are using, say for example, organophosphate, and then we are using pyrethroid. So in the same area, different houses, we are using different insecticide. It's little cumbersome for planning and implementation purposes. But yes, it has been recommended, and in some places it is done. It has been proved very nicely. There is another example which will have very clear picture. If you see the mosaic approach, this is one village where one house we are using one insecticide and another house we are using another insecticide. This is very cumbersome, but this is a recommended and proven procedure where we can have one option to delay the insecticide resistance precipitation. This is slightly, one is little, one is, this is all fine, very fine type, that in different houses you use different insecticide. But there is nearby villages, you can have one village in one area, because basically a group of villages are taken as a unit in a health sub centers or in a primary health center. So if you take one village with one insecticide and one village with another insecticide, this is another option for the mosaic spraying. This is again for the cross border area, or some area where you have very compact and you do not want the mosquito to come here and there, or the resistance. And this will have another advantage, that if one insecticide is used in one village and another insecticide is used in another village, if the mosquitoes are left over and they are migrating, they will be exposed to another insecticide and they will be killed. This is another good advantage of such kind of things. Then comes the mixture. Mixture, there's a good example that LLIN, now the PBO nets have come. You are using synergist, non-pyrethroid or any synergist on the rooftop. And the whole, what is the basic concept is that the mosquito will try to come initially, if you see the habits, I mean, people they have studied, they try to come on the top or they try to come on the bottom side. So what happens? All the four pans you have, you use the nets are with the synthetic pyrethroids, and then the upper portion, the roof is coated with the PBO, or the tugging portion has been coated with the PBO. So what will happen? And basically this has the whole philosophy has been taken to put the rooftop with the PBO, that when the smell comes, the fume comes, the attractant, basically carbon dioxide, mosquito will try to come and pull. So they will be killed. They will get exposed in a different system with one or synergist, non-pyrethroid, or when they try to come here for biting, they will be exposed to the pyrethroid. So this is sort of in one place you are doing mixture. There are another examples where you put two compounds, insecticides, and then try to do the spraying. Then there is another, the second aim was overpowering the resistance by continuous killing. How continuous killing of the resistance mosquito is basically unrelated insecticide you mix. That is what I have shown in the mixture formulation. So basically both the objects, either you rotate and confuse the mosquito, or you forcefully you kill the mosquito by two insecticide pressure creating more. You have to kill all the resistant population so that no survival of the resistance and further no precipitation of the resistant population comes on. Having understood all these things, what is the summary for the resistance management? Basically, best is prevention. That is right from the beginning, it is best. But if we want to wait and get a proof for the level of resistance, what exactly are the evidence of our tool has failed, that is not advisable actually at this moment, when we, especially for malaria, we are targeting next another 22 is gone, now another 8 years for, and basically 27. So only 5 years left. 3 years, last 3 years is for the sustenance period. So that is not advisable. So best course is IVM approach is recommended, where one judicious use right from the beginning, which is very, very critical, and many of the countries, where are the regulation, where are the policies, what is the monitoring of the doses, because all these human error factors have facilitated also insecticide resistance in addition to the exposure of the insecticide. So basically our insecticide resistance monitoring plan has to be, because otherwise there will be no evidence generated. Once there is no evidence generated, there is no insecticide resistance management plan. So basically insecticide resistance monitoring plan is the prerequisite of insecticide resistance management, which involves situation analysis, regular entomological surveillance, which just now Michelle also has emphasized, and resistance monitoring with the data management, coordinated effort with the research institutions, resistance mechanism, capacity of the national program, and then advocacy, so that the resources are not curtailed even at the time of low number of cases, because people will think that the disease is not there, so what to do with the mosquito? So that should not be done. And logistics, yes, definitely logistic has to be maintained. Now the example, when we started with this background, we started working for Bhutan, where they are just nearing the elimination target. 2019, only 2 indigenous cases were reported, and their target is by 25, like next 3 years. So if you look on the Bhutan, before we made the insecticide resistance planning, we wanted also to have a whole background. So DDT was replaced with deltamethrin in 1995. They started also in early 50s. 1995, DDT was replaced with deltamethrin. Then 98, ITN was introduced. Then Bhutan has started indoor residual spraying only for outbreak and focal in 2003 with the deltamethrin, and then other pyrethroids, and then started LLIN in 2006. So this is the background of Bhutan, how to make the IRM plan. That is why this talk I have taken. If you see the Bhutan, they have done the mapping very nicely. They have done the mapping. You see the down below area. It is clearly divided into 3 areas. One is high altitude area, where basically there is no risk for malaria. Then there are green color area. This is potential risk, because this area is actually receptive. But this area down below, this is the low risk area. This is some of the portion is bordering with India, and then China and other places. So this is in northeastern states of Assam. There are 2 or 3 districts where they are bordering. So this area they have divided into low risk area because of some migratory population and some indigenous cases they have reported. If you see the entomological surveillance, basically the presence of the data, they have mapped their districts. These are the 7 districts, 1, 2, 3, 4, 5, 6, 7, and 8. But this is, so this 7 or 8 districts, they have mapped that what are the vector species they are found. And basically they suspected Pseudovilmeri. This is the Culicephasis also they have found, Fluetilis also they have found in different places. But you combine both these maps together, this is all the part of the strategy when we started working on this. If you see the map, this area is low risk area, I mean malaria risk area for Bhutan, and this area they have got the Fluetilis, Minimus, Dirus, Decilatus, Japorensis. But in present study, last decades, they have not found these vectors. What they have found, they have found only 1 Dirus larvae in forested area in Sarpang district here, this one. So, and Pseudovilmeri, they are suspecting as a vector along with the Culicephasis. They have done the tube test and the CDC bottle assay test. The CDC bottle assay test, they take the help of the international agencies, WHO. tube test they are doing. How they will do these 2 tests for the program, how they will do, the whole plan was made for them on this issue. But before, they have to set some timings. So what happened? There was one hypothesis which we started working on that. And then when we discussed, then the resource constraint which it emerged, and then we have little more compromised. So how we have done. IRM endemic area was the first task. It was explained, then data analysis, then decision and management based on WHO guidelines, whatever is feasible for their area, because they have got very less number of cases. And this is very important that Bhutan is very keen to do resistance monitoring and do the resistance management, even though the cases are hardly 22. So the talk came up that sentinel sites has to be managed. Now, how to do sentinel sites, what frequency it should be done. There was a planning challenge that because of the altitude area, they will not get many of the species for conducting the test. They have different seasons where extreme cold, similar November to February, they cannot go anywhere. So this will be highly cold climate, they will not get. So the team has to go and stay for some more time, devote more time. And that is all is part of the planning of the whole management issue. So the first part was made, plan was made like this, that they have got districts, Jiwang, and then Chiwang, like primary health centers and sub-district Jiwangs. So what happened? The plan was made that 2 sites will be taken from all sub-districts in 7 malaria endemic districts. They will be identified, segregated, and then there will be, down below, they will be tested for adult susceptibility. If they don't get, then the larvae will be collection, egg laying, and then F1 generation, and all these things will be done for this. But what happened when the discussion was, it took little more progressive, then it was seen that they don't have so many power. So what happened? There was little compromise formula. One district, Sarpang, the central one, that was that there it will be taken 4 sentinel sites, but rest all the districts only one sentinel sites from each sub-center area will be taken. That was the modification, which was a compromise formula, which was made. Rest of the things, when the mosquito collection and all those things. So these are the things which program has to always take a call on these issues. Now, so that is what is described here, that based on the mosquito collection, one village, first they will do the primary survey, preliminary survey, and then they will be fixing the sentinel sites, one village in each sub-district, and then like that, 8 sentinel sites they'll be doing. Now, the problem came when the entomologist, one entomologist, insect collectors, they will have to be sensitized, they know where to go, how to, what is the plan, transportation, then they have to do what kind of collections, which kind of mosquitoes. So Monday to Wednesday evening or Thursday will be approved, because if they go and they have to stay for that, then one day they will go, they will stay. So the whole detailed plan was made for a week time. So if they go on Monday to Wednesday, they will do. Suppose they go on Monday, they will start working on Tuesday or Monday night itself, if they reach early, except one district. So the whole detailed plan was made on that. And these are very important for the program people. This was the plan which was made for the frequency based on the seasonality, that these districts they will be covering in June, this will be in July, this will be, because this whole one from the team from the central only was available, made available. So many entomologists and so many insect collectors in each districts. So June, July, August, September, like that it was done. So the chalked out and calendar of activities was made. Sorry. Now the issue came that as I told you, the district, they don't have the trained technicians and capacity to do the sentinel sites also, can be 3-4 hours. So accordingly, the central team was brought in picture, and they will provide these insect collectors and human resource. So they will spend 4 or 5 nights. So like that, issues has been resolved in the whole plan. And that is what is required. Now the problem came that which site should be, where from? So basically they were explained what is the zonal concept, what is the zone and what is the district, IRM unit, IRM zone, implementation unit for IRM, that will be sub-district. And then while going for sentinel sites, initially it was thought that one each from PHC, but later on only one village in one sub-district will be selected. So this whole concept was explained to in the document. So this is IRM zone unit and implementation unit and sentinel sites was explained to them. And then for general information, this was also explained that there was a question I was just going through in the chat box also, that what we should do. See, this is the basically the whole plan. What if you are doing resistance and you are getting you want to delay the resistance? First you do the baseline either by this tube test and then susceptibility test. Check the susceptibility. If it is you are finding no change in the susceptibility, continue with current strategy, whatever you are doing the vector control operations under IVM. But if you get the high level of survivor in the test or a small increase in survivors, then what you have to do, if you are getting a small increase in survivors, you have to increase your monitoring, rotate the insecticide to resolve the pressure. But if you are getting very high level of survivors in the test, then you have to repeat the test, and then you have to go for insecticide class, existing whatever insecticide class you are doing, then change the different insecticide if it is available. That is a big question mark. So these are the options which have been given. There is another example I have given. This is also, if the whole product fails, what you have to do? If the product is not coming in the quality, first you should suspect that whether it is a human failure or it is the, before you suspect technical failure on the problem, you must try to see is there any logistic. So please do not panic on that. If resistance declared indicated, try to analyze whether it is a patchy spraying or suboptimal spraying. Instead of blaming the insecticide, instead of blaming the whole team, try to analyze these situation and then try to do it. But if yes, if the resistance is confirmed, then you have to go through, check this, why it has in reapplication, and then you have to do, either you are doing for indoor residual spray, you have to check the equipment, coverage, insecticide, same thing, larvicide, same thing for bed net. All these things ensuring, then only you switch over on the next insecticide. Just a glimpse of the after the Bhutan, one last slide for the India, how the resistance has changed. I have given only one vector because India is in the grip of 10 vector complex, primarily 9, 10th is also suspected. Not suspected, it has been basically confirmed in many places, but it requires whole country multi-eco-epidemiological situation trials. It is basically on major vector culicephasis, how the resistance has started coming down, and then again the resistance against all the 3 insecticides, red color mark, if you see the last one, it has increased. So and that is also a core malaria transmission area. So basically, my message is that if you are doing insecticide resistance management, I have just flagged certain issues, and these are the basic challenges for any large country when we are talking of insecticide resistance management. That if insecticide resistance is evidenced by tube test, intensity bioassay, data analysis, then whether it is patchy, then dedicated entomologists are there for data analysis, because data analysis is very important issue. If only malathion or synthetic pyrethroids are available, we don't have many insecticides in the pocket. Our basket is very limited. Insecticide rotation challenge is basically shelf life. If you are getting shelf life of 2 years, how can you recommend rotation in yearly basis? Number 1. Number 2, transportation time. If you are procuring insecticide of 2-year shelf life as per the regulatory instructions, then it will take some time for reaching to the consignee, from the manufacturing site to the states or the district, and then at the site where the spray is required. So before this shelf life, you have to consume it. So you have to think of all those things. Then the very important point is that pyrethroids. Synthetic pyrethroid, because dose is not same for all the synthetic pyrethroid. Some delta methylene like 20 milligram per square meter, and for alpha cypermethylene or others, lambda I think is 25 milligram per square meter. Now, if the dose is not similar, you have to take a factor of comparative to evaluate. And if you are, there is one option, you divide the area and accurately you require which product you want, which brand you want. That will be against the rule of the procurement in principle. So you, when you make a general requirement of synthetic pyrethroid, the delta methylene will also be bidding the pyrethrum, and as per the rule, lowest bidder will come. So how to rotate, that is very, very different, because every year, suppose the same per company, same product comes as the lowest. This will be another challenge for insecticide resistance management. Then capacity, logistics. Immediately you have to go for lambda, you have to go for delta, you have to go for DDT, you have to not, DDT is now out of question. You have to go for organophosphate, and carbamate has not been recommended. Now we are actually, I was surprised to see the previous presentation that even carbamate also Aedes aegypti has shown resistance. So this is a very burning question. So these are the issues and challenges. I'll stop here. This was one aspect which was taken for Bhutan, because the area is limited, the work is also limited, not in as compared to the Indian system. Thank you very much.
Pradeep, excellent presentation as always. So, you know, it's like listening to the professor. It's all those decades of experience that you bring to bear in your presentations. It's not book learning. It's experience and insights, understanding that you built up the long way. And you can talk of experience when you teach us these lessons, because that's basically what you've done here, is to remind us of all the different factors involved, what should be considered. So again, a masterclass there. Thank you so much, Pradeep. I think everyone thoroughly enjoyed that. So we come now to the question and answer session, and may I again express my sincere thanks to all three speakers. Wonderful presentations. I think everyone has really benefited, learned from, and enjoyed the presentations. Really great presentations. We come to the Q&A session. A reminder to our audience, please put your questions box, not the chat box. We're only going to be monitoring the questions box. So let's go to the Q&A box. I think the first one was from Stefan Carl. Yes, and the question or the comment really was addressed to Nigel, to you. Stefan says the biting behavior that we observe in PNG today for Anopheles ferrati resembles the before DDT profile. So I also find that fascinating. You know, DDT resistance must go back, way back. And it seems in some areas there's been a reversion to the ancestral genotype, or reversion to susceptibility and so on. Would you mind just spending a minute or so, Nigel, on this issue of, if you relieve the insecticide pressure on a population, is there a trend whereby some insecticide classes will revert to historic susceptibility quicker than some others? and also the issue of, toxicity susceptibility does not mean the same thing as behavioral, the intensity of behavioral shifts or so on. So while you may get reversion to ancestral type, if you like, in toxicity, there may be a different rate of reversion or retention of behavioral changes and so on. Just this interplay of those factors, if you wouldn't mind just giving us a few pointers, Nigel. I know it's difficult for you to say.
Thanks, Leo. Look, seeing the phenotype changes, we're seeing them quite strongly being affected on islands. Islands have a small population size of mosquitoes, so a change in a population on an island may be an easier thing to achieve and maintain. Where Stefan is seeing in PNG, the Anopheles frowdi population is enormous in PNG, and it exists in lots of places where there is no malaria control. And so even though there might be incredibly strong selection for early evening biting, there's gene flow back into that population from outside of that population, reverting back to its native genotype. I mean, I think we're seeing these phenomena happening on islands more so than we may be seeing it in PNG. But that would be my only explanation. I couldn't tell you about one insecticide over another. What's happening in that population is that the mosquitoes that are biting earlier in the evening are getting their blood meals, they're making more offspring. If that early evening biting behavior is genetic, that'll be pushed on to the next population. It's interesting, in the Solomon Islands, we did some mark release recapture experiments where we collected late night biting Anopheles frowdi and early evening biting Anopheles frowdi, and then we marked them with dust and we released them, and we saw that the early evening ones we caught bit both late and early in the evening, and the late evening ones we caught bit both late and early in the evening. So there's a lot of plasticity that seemed to be going on. And also a large, part of that early evening biting population were nulliparous. They were quite young mosquitoes. So I think seeing those phenomena on islands where you have an isolated population with limited gene flow into it is easier. But in PNG, you might be fortunate enough that you won't see such a dramatic change in that biting all the way through the night and then to biting early in the evening only.
Wonderful. Thank you for that, Nigel. Yeah, I mean, that makes perfect sense what you say there. On an island, you have a limited population and you can maintain that pressure on that population, whereas in a mainland or large geographic area situation, there's continuous integration or inflow of the ancestral genotype or genes. So it all makes perfect sense. We have another question, and I think it's more appropriate for you, Nigel, from Agye, if I get the name correct, and it's about alleles and so on. So, we don't have much question and answer session time. So I wonder if I could ask you just to quickly summarize that in the chat box to Agye, Nigel, and then I'll move on to the next question.
Okay, could you read it? I'm just trying to find the question.
Oh, I'm sorry. It's the second one after Stefan, the very top one. If you scroll up to the very top.
Okay, I've written an answer into that. This is one about flooding in Queensland.
No. It says, hello, this was a very interesting talk and I really loved you for that. Thank you. However, please, I did get the molecular aspect well. Specific alleles for this resistance.
Okay, look, that is the $1 million question, isn't it? We think it might be driven by circadian rhythms. There might be a whole set of gene families that may be involved in this. Look, that's like looking at the needle in the haystack, and we can now that we have genomics tools, we can actually potentially select for early evening biting populations, cross them with late night biting populations, and look at the activity of next generation. It's a huge job to do. So the quick answer, no, we don't have really any idea, but there is a way of approaching that. And then what do you do after you work out what those genes are? What's the practicality of that?
Yeah, thank you for that, Nigel. Thanks for that. Okay, then we come to Mohammed, your question. Pradeep, can I ask you to try and respond to this one? I mean, this is it's a situation you don't ever want to reach, and that's why we do insecticide resistance management. What should we do when resistance is confirmed against all 4 major groups of insecticides? I mean, that's precisely why we do IRM, is to avoid reaching that stage. But let me not answer on your behalf. What would you say to that?
The basic answer is last but one slide, that IVM is the basic answer. If you have got resistance, confirmed resistance, that is after intensity biopsy also. So far, the situation has not that all the 4 class. I mean, we just saw one glimpse in Aedes aegypti in one slide, but in Indian scenario, yes, it is not there. We have very limited number. We have to go for judicious use of insecticide and rotate the group so that the mosquitoes are diffused. Now the choice depends on which group you have in your basket. You have got chlorinated hydrocarbon, DDT is gone, but you have got organophosphate, you have got carbamate, you have got synthetic pyrethroid, you have got in synthetic pyrethroid, you have got 5 or 6. You have got some new molecules also which has coming, and WHO has just now released the list how to do the susceptibility test of some 6 or 7 new compounds which are already approved. Some of the countries they are procuring, some of the countries they can do smaller area, you can just go on rotating. But for larger country, larger issues, I have just shown. So IVM is basically you have to judiciously use, area is specific, you have to see. And basically you don't have much choice in LLI, also it is only synthetic pyrethroid as on date. There are some issues related with behaviour, that's also, and new insecticides class is also coming, neonicotinoids and other things. So we have to have certain things, but all depends on in your country what has been regulatory bodies they have approved, whether that has been tried and they are ready for use in the program. Thank you.
Okay, so there was a comment here by, thank you, Pradeep, thank you for that. Good, again, a lesson from the master there. Raja Lakshmi Anbalagan, yeah, that the same resistance that you picked up in PNG, Michelle, in the dengue vectors, Albopictus and Egypti against DDT, has also been picked up in Tamil Nadu in India,
Yes.
and they're planning to extend their work and target site identification in dengue vectors in Tamil Nadu. So just a comment there. I don't think there's the request for an answer to that. Wilfredo makes the comment, rotational use of unrelated insecticides is recommended to relieve the pressure for buildup of insecticide resistance. But currently most insecticides in use are under the group of pyrethroids. I'm assuming this is for impregnating bed nets particularly, and very few under organophosphates, carbamates, and organochlorines. So yes, I think it's again more a comment rather than seeking a response.
Right.
There is, of course, that whole new generation bed nets being coming out and being endorsed by WHO, principally being taken up in Africa. I'm not aware of significant uptake in Asia Pacific, but those are becoming more and more available. So, you know, there are pros and cons, and in areas, much of Asia, still a good level of susceptibility to the historic pyrethroids. I think National Malaria Control Program with using what's available. Do you want to make Pradeep, this whole issue of, yeah, you respond.
No, you are right. You are right. Basically, I mean, what you have told is exactly, we don't have much of choice, basically. That is the limitation what we are having. New compounds, as I am been asking, that it all depends that whether it has been with your regulatory body in your country, whether this is all ready for trials and uses. If we cannot use, so our basket is very, very limited. That is the basic core crunch for this program, even for LLIN or it is for the IRS products. There was one product which came, but suddenly nobody is interested in that. Long, because one round only will keep long persistence. Basically, what we aim that kill all the heterozygote population.
Thank you, Pradeep. So here's another one from Gaurav Kumar. Dr. Pradeep, in view of rising insecticide resistance in the major in Anopheles calicifacies, again, classes, organophosphates and organochlorines, what should they do? I think you've already alluded to that in your presentation. It's integrated vector management. You know, it's those different strategies that you. And I think, Gaurav, if you wouldn't mind, I'm trying to save time here. The link in the chat box to that new Bhutan insecticide resistance management plan. And I'm sure there's detail there
Yes.
that would guide one to how to respond or avoid build up or having to reach the situation where you have full-on resistance. So, you know, taking your way around insecticide resistance manifesting itself. Where am I now? There's a question here from Dr. Kalpana Barua. Hello, Kalpana. Good to see you present. Kalpana says, Michelle, nice presentation with high insecticide resistance in both Aedes aegypti and Albopictus in PNG. What is PNG using for Aedes control? What do you do about for dengue control and specifically dengue vector control? What is your primary approach to dealing with dengue? I'm putting you under the block there, Michelle. Please try answer.
Thank you for this very, very important question. So in Papua New Guinea, especially in terms of dengue control, we don't have much information because most of the control measures, especially in light of dengue epidemics, are conducted by either provincial health authorities, or we also have cases where mining or agricultural or other entities, they conduct small-scale control of mosquito vectors. So really, I cannot really tell you exactly what vector control interventions we're using, but I can give a general overview of what people, these small-scale control programs are doing. They're mostly using fogging and IRS, especially within mining areas, to control mosquito vectors, and that covers also the dengue vectors. But in terms of a national dengue vector control program, we do not have, I believe, a program in place at the moment.
Michelle, before you go away, I'm going to jump one question because it's related from Oluwafun Milola. Thanks for your presentation, Michelle. From your research, you noticed a high rate of Aedes resistance in PNG. What could have caused this? And I'm assuming it's just reliance on one or two products and persistent use of those one or two products. And one is kind of forced in that direction because if you're doing IRS or so on, you have to use products that are not dangerous to babies and humans and so on. But okay, you answer the question if you don't mind, Michelle.
Yeah, sure. So I think there are factors other factors involved that could be, contributing to these increased vector resistant profiles that we're seeing in Aedes. Especially with recent data from Tom Smith, as I presented in the presentation, showing it in particular to Aedes aegypti populations, the Cadia mutations that we now see are imported from other countries, rather than these mosquitoes naturally evolving these resistant mutations to the insecticide selective pressures on the ground. So with these imported mutations, whatever insecticide pressure that are done on small scale, and particularly for Aedes aegypti within urban areas where there are, companies and provincial health authorities using insecticides to control these mosquito vectors, that selection of the mutation is developing and it increases, and this is what we're seeing now, resistance to the different pyrethroids and
Thank you, Michelle. Yeah, interesting that, that if, imported mosquitoes are contributing to the development of resistance, it suggests significant importation of such genes for it to have an impact on the local genotype. So very interesting. There's a comment here from Vimal, Dr. Pradeep, for you. Does
Yes.
India plan to use any new class of chemicals other than the organophosphates, organochlorines, synthetic pyrethroids in vector control, neonicotinoids, for example? Any comment on that one?
There are some, Leo, there are some products which are under trial, and basically the whole policy is that the trial has to be completed, whether it is even the WHO has given other thing like clothidine and flypyridine and then transflothine, but the trial of new product, whether it is a neonicotinoids, anisaids, it has to be completed for as per our common protocol, and then it has to be approved by the regulatory authority. Then only product can come in the program. Program does not per se starts the trial business of its own. When manufacturers has to make the product and come and request the program for inclusion, then only the whole procedure starts. That is why IVM becomes more important, judicious use of available insecticide within your basket.
Sorry, coming straight back to you, Pradeep. I come across this question every once in a while, and it'd be interesting to get your take on it. So when field entomologists go into a particular area and they want to collect wild material for susceptibility testing, you get a mixed bag of specimens and species. And, there's always the challenge of getting adequate numbers of replicates for statistical purposes to interpret your assay. So, how does India deal with that? You go into a particular village, you come up with relatively low numbers of mixed sensulatum species on top of that. So, your response.
Yes, very important question. I will not say it is $1 million because we have been doing this with our own hand in last 20-30 years. See, if you are not getting adequate number, first is that you are not reaching to the proper place where mosquitoes you want to get collected. Number one. Number two, there only that is the need of the entomologists and vector control specialists there, which are lacking. Slowly, whole world it is diminishing. The third one, we have experience only in one species, minimus, in northeastern states where adequate numbers are not there. The question is, when you get mixed number of mosquitoes, if a proper trained entomologist is there, he will identify. If you are not having adequate number, even after having a trained entomologist, then you cannot perform the test without adequate number. You have to have species wise, then you have to bring it to the laboratory and go for F1 generation, you can take the help of research institutions. When you have like minimus, you have got very, very less numbers. That is true. So you have to depend on the laboratory rearing and then getting F1 generations. You can't have 5 numbers and 10 numbers, because if you see the calculation formula in one of your APMEN talk, I have explained it, that 5 to 20 mosquitoes death, 5% to 20% death. If you are taking 5 mosquitoes and one is dying, so it is 1 by 5. It is 20%. You have to discard the test. So less number of mosquitoes, it cannot be. Species wise, different susceptibility test has to be interpreted. Otherwise, there is no question of mixed population putting in one tube and then getting the test. Against which vector you are doing actually?
Thank you, Pradeep. So Prudence, I'm not sure if you're still with us. Hopefully you are. I'm going to start jumping because we're running out of time. So I'm going to be more selective. I'm not just going down the list of questions. And my apologies to the audience, but we are running out of time. We're almost at 2 hours. And let's try and, okay, I see. Okay, so I'll get to those who raised your hand. Hajira, let me write that. So, Michelle, let me direct this one to you. I don't know how effectively you can, but let's hear your response anyway. It's always good to hear from people that have spent time in the field and work with communities or so on. So, okay, here you have now concrete evidence via courtesy of Nigel, who's done the genetic studies, and that's another question that was asked. Anyway, that there's been a shift to early evening biting outside of the hours where you're protected by the bed net. So now you're sitting outdoors around your cooking fire and so on. What do you do about it? What do you tell those villagers? Look, you're no longer being protected by by the bed nets. Those mosquitoes are now targeting you here exactly where you're sitting outdoors. I mean, that's the $1 million question. What do you do about it? You know exactly where the risk is. So if it's outdoors like that, what does one tell people? You can tell them use repellents, but it falls on deaf ears by and large. So does anyone have any suggestions? Michelle, Pradeep, Nigel, anybody in the audience?
I can suggest what we are going to suggest for the program also. Outdoor transmission, you are very right that it is $1 million question. And IVM is not only use of judicious use of insecticide or LLIN or larvicide or bednet. It also incorporates community involvement, community engagement. Community has to be prepared to protect themselves also. If they want to die, nobody can help. But the role of the program is public health program. The role of public health program is to have that the disease is not a major public health problem. So the people who are living, if there is a shift in mosquito, that again there will be a need. There only the entomologist only it can solve this issue. They will study, they will fix out the localities, and then they will try to convince the residents, please do this business, try to put, I mean, if the mosquitoes are changing behavior, we have also to change our behavior. So there has to be sync between the behavioral change patterns. Otherwise, go on doing spraying, go on doing. Similarly, like larvicide, we have been talking in dengue. This is the health department becomes a victim department. You cannot, you go on creating the mosquitogenic condition and you go on putting the larvicides. This will never end. So some responsibility and accountability has to be fixed.
Yeah, easier said than done, Pradeep.
Yes.
I take your point. Yeah. Michelle, did you want to respond?
Yes, I also agree with Dr. Pradeep with like community participation. This is really important, especially if we have early biting and vector species and they're not, the community is not protected by the intervention tools that we use, which are mostly indoor intervention tools. Here in PNG, however, we're now working on a new project, another project that was data that was presented in previous APMEN presentation. We do now understand and realize the importance of targeting these outdoor vectors as well. And again, instead of not only, as Dr. Pradeep said, it's not insecticides only that we look at to rotate or management. We also look at the different tools targeting the different areas of habitats or different areas of where are gathering where they are and vectors are coming in contact with them, and to try to protect them as well. So in particular, outdoor tools, we're now looking into that and trying to evaluate that in PNG setting where most of our primary vectors are outdoor biters. So in terms of relaying whatever information we have to the communities, I mean, we do tell them that, okay, the mosquitoes now have shifted the biting, and you are unprotected at this time in the evening. However, like it's easier said to have community participation, and most of these villages where malaria is really high, especially in PNG, human behavior, we cannot really go there and tell them to change. It is very difficult to convince them to change from generations of lifestyle and behavior that they have in terms of their daily livelihoods. So I believe we could have community participation, but also as entomologists, what we understand now, we could recommend appropriate tools that would target these different areas that are not targeted for vector control at that time. Yes.
Yeah, so I don't want to dwell on this too long, but it's a very, very complicated, complex issue. And of course, fully agree with you that one has to, we haven't been doing very well with community engagement. We can improve a lot in our messaging and explanations and so on, and getting communities to understand the challenges and risks and risk reduction behaviors and so on. But what exactly are the tools that one tells the community they must now adopt outdoors to overcome the lack of efficacy of the bed net, which is no longer protected? Impregnated materials, your repellents, etc. If you use that, you have to go and pay for it yourself. So communities don't have that much disposable income. So by and large, we're not easy for communities to address outdoor biting issues. And I'm not trying very hard in doing trials and research in trying to find a solution or at least an abatement approach to reduce that outdoor risk. But we need to do better, really, especially in Africa. This is not a problem that's going to go away by the end of 2030 as it is in Asia Pacific. Africa is going to face a much longer uphill battle. Thank you for that, Pradeep and Michelle. Fully agree with you. We need to engage with communities and at least tell them about risk and try and help them find ways to compensate, somehow reduce that risk behavior. Nigel, I wanted to ask you, somewhere in the list here, I missed it somewhere. There was a question, your bar graphs, different biting, nocturnal biting behavior. That based, was that CDC light trapping, human landing catches?
Which bar graph were you talking about? In Queensland study?
Well, generally, but okay, respond to, you know, to the trials that aware of. I think for different species, there were bar graphs of.
Oh, that was yeah, that was John Kevin's work. I think they were based on trapping and then changing the trap every few hours.
Okay, so it wasn't human landing catches, it's more CDC light trap type catches.
Yeah, let me go back and
No, I fully understood. Okay.
But those windows in time to me look like and I've forgotten it from the paper because
Yeah, fair enough, no problem. Thank you for.
But there was a question about human landing catches versus trapping, and we have in Queensland done both with the Ferrati population, and we've got a CO2 baited fan trap that if we change the capture unit every hour, you can get similar results. You don't get as many Ferrati because human landing catch is the best, but you still can get those profiles without theoretically.
As long as you get the same trend. Yeah, the numbers may differ, but as long as you get the same trend line, they reinforce each other. That's important.
And we've done that work where we've compared human landing and traps, and you do get the same outcome.
Thank you, Nigel. So let's get a bit more live interaction here. I can't see Wulan, Popo, Dr. Borne. Can you see any hands raised? I think Hajira.
I did, and I allowed Hajira to speak.
Okay, Hajira, please ask your question. We'd love to hear from you.
Yourself.
I see there are two other participants.
Maybe we can go to Marina Pangabian.
So if you're able to unmute yourself, any of those, just talk to us. We're all ears. While we wait for that, so any one of our esteemed panel members, here's a question from Bala Muhammad. It says, with the changes in the mosquito biting behavior, can millions of IRS LLINs be the solution? Intuitively, if you ask me, no, it's not going to help. You know, it's the effect. Whatever chemical you are using is having the same impact on favoring those with a gene that exposes the population to bite outside of the insecticide pressure period. So any response from anyone? Does it help changing insecticides?
No, look, it won't. You're targeting both indoor biting with both of those interventions.
Yeah.
And they basically similar effect. Just going back to that question you asked earlier, I just looked it up. So John Kevin's paper, they use barrier screens and human landing catches.
Okay. Thanks for that. Thank you, Nigel.
That's okay.
So Hajira and the others who raised their hands, Wulan, do we have a technical problem or is it just they're not Yeah.
I'm not sure, maybe they have internet problems, so they cannot unmute themselves.
They're not able to unmute themselves.
Yes.
This is the problem. Yeah.
I think so.
All right. I don't. Mohammed Asif Mahmood asks here, in case of less number of, can we expose required number in different occasions and then combine the results? It goes to that sample size issue again, Pradeep, where you go into a village, you do a human landing catch, you get very few specimens. So the question is, can you
Repeat it regularly on daily basis.
Yeah.
That is what the answer in WHO manual, recent manual also it is given, and we have been doing the same thing.
Yeah.
That if you don't get one day, so you stay little more longer time in the same area, collect nearby area and do that. That is where the concept of IRM zone and IRM sentinel sites and the village comes. Yes, you can stay little more longer, collect on daily basis and do. If on the same day you cannot do the replicate.
Okay. So I just want to remind our audience, please, at the end of the webinar, there's going to be a very short poll, and we'd be very grateful. It'll take 1 minute of your time. Just fill complete the poll if you don't mind. It helps us. So we've been going for just over 2 hours now. I think it's unfair to keep the audience up for much longer. So to all our panelists, Michelle, Nigel, Pradeep, in alphabetical order, what a wonderful webinar. We say genuine, sincere thank you to each of you for your time, for your wonderful responsiveness to questions, great presentations. Thank you for keeping the audience interested, and we look forward to seeing you part of our next webinars coming up. Thank you, everyone, our respected audience members and our panelists. Thank you so much. Have a wonderful day.
Thank you, Leo.
Thank
Thank you.
you.
Thank you,
Thank you,
Leo.
Dr. Braack.
Thank you. Thank
Bye.
you.
It was nice meeting all people. Thank you.
Yes,
Bye.
you, Pradeep and Michelle.
Thank you. Thank you, Nigel and Michelle. Thank you. Bye-bye. Bye.
Thank you. Bye.